So we'll have a quiz on Friday. We have an exam next Wednesday. Yay, exam! We'll have a help session on Thursday. Let's see. There's so much in this class because we've got the lab too. Help session on Thursday at 8:30 p.m. on Google Meet. Let's see. I'm available to help Monday, Wednesday, 1 through 5 p.m. Although today I'm only here till 3. Okay, so sorry about that. And then let's see. Oh, your lab report. Lab report. It could be due Thursday.
That works.
Okay. Lab report due Thursday. Yeah.
Inquire.
Okay. I'm trying to think of how it relates, but I'm not going to spend much time on it. Okay. So, am I missing anything we've got going on?
We got some chem events this week.
We have what?
Chemistry events.
Chemistry events. Oh yes, it's chemistry week, right?
Yeah.
Yeah. So we go to the chemistry events. There's a poster session. It counts as an epic, right? You can look at 3 posters, write about them.
There's one Friday at 4.
Yeah. Okay. Do you want to advertise? You're gonna do a better job than I am.
There's one Friday at 4. We're gonna do some demos and decorate for spooky stuff and have food and then there's also ethics this week, which are informations are on the posters that I don't know.
Yeah. So chemistry week. A lot of fun events. Okay, so let's where were we? Where were we before we were interrupted? Remember we were talking about hemoglobin last, and hemoglobin has 2 different states, and these states are based on the quaternary structure of hemoglobin. So we have a T state, which what do we know about the T state?
It is looser.
Okay, looser or
Doesn't
weak binding of oxygen, let's say. And oxygen is always O2 for us, or here it's going to be O2 for us when we talk about it. And then our state is then strong binding of oxygen. Or we could say affinity or whatnot. And we need these 2 states because what protein is hemoglobin? Hemoglobin is an oxygen transport is the important part of that protein. All right, so in other words, it has to pick up oxygen and it has to drop it off. So you want it for it to best function in your lungs. It would be in the R state and pick up oxygen. Let's see, the R state is closed in. It'd be the R state and pick up oxygen, and then in your tissues, it would then go into the T state and release oxygen. And we can see the benefit of that if we look at this graph we looked at last time, that what we have is if we look at a hemoglobin, this represents the binding curve of hemoglobin that is changing states, okay? And so if we look at the oxygen in your lungs, that's about here. Okay, that's the oxygen concentration in your lungs, we could say. And then in your tissues, the oxygen concentration is about here. And so I've done this kind of poorly, but it looks like we get about 0.35 or 35% of hemoglobin's oxygen binding capacity. So we think about the math we've talked about in class. Your book likes to use Y instead of theta for a fraction of hemoglobin sites bound. Does that make sense? So you load up to about 100%, maybe 95%, and then you drop down to maybe it's not exactly 35%. I'm just pulling that number off a graph. I'm not doing it from memory of reading it somewhere where it's perhaps more accurate. And this will be different for each person, how much you actually pick up and drop off as well. Questions? Okay, so by changing states, we get that. I think if you look at the high affinity state, you can see how bad it would be if it just stayed in the high affinity state, right? You'd load up to 100%, and then maybe in your tissues, you drop off 1% of your hemoglobin capacity, if that. Right? So you see how it's good to be able to change these states? All right, we had three substances that we talked about that helped change the state between the tissues and the lungs. What were those three substances? Okay. That I'm going to say help hemoglobin change to the correct state. And so I heard an oxygen, right? So oxygen binding favors the what state? The R state. So it's like a rich get richer situation, right? The oxygen binds, that makes oxygen better at binding oxygen. That's what you want in your lungs. There's a bunch of oxygen. You want to pick it all up. What's another one of these? Okay, carbon dioxide. Okay, and that favors the T state. All right, and that makes sense because in your tissues you are producing carbon dioxide, and that's where you want oxygen dropped off. So there's more oxygen in your tissues. That helps hemoglobin to switch to the T state and drop off hydrogen or drop off oxygen. What's the other one? H plus. So H plus ions. All right. So pH, right? And they favor the T state. All right. And we know metabolism makes your blood more acidic. And your blood is more acidic. It's about pH 7.2 in your tissues. Kind of depends on what's going on as well. But your blood is more acidic in your tissues and it's less acidic in your lungs. If it's more acidic in your tissues, you have more H plus ions, you favor the T state, and hemoglobin drops oxygen off. All right? And then the Friday before break, we went into gory detail, right? About, especially for these two. Especially for the last two here, we went into gory detail about how that works. And so you're responsible for giving molecular level descriptions of how these two things, how all three of these things, but really carbon dioxide and H+, that's what I'd really be interested in. What's going on with protonation states and how does that change the charges to make hemoglobin change shape? Okay, remember it's all about charges, or mostly about charges at least. All right, I'm not going to go over what those are again. You can go back and look at the lecture, look at the notes. What I do want to do is I want to talk about our last molecule here, which is 2,3-bisphosphoglycerate. The name's there, saving me from having to remember what it was, which is nice. Notice this is a very negatively charged molecule, right? Okay, this is a small molecule. It's hanging out in your blood. And the amount you have in your blood depends really on what altitude you're at. And it's going to not change its concentration based on where it is in your blood at all. Or it's not going to influence, it's going to globally change hemoglobin and favor the T state. So the binding of 2,3, and I'll even accept BPG, to hemoglobin favors the T state. All right, let's talk. First of all, just take my word for it. All right, and so at sea level, you'll have a certain concentration of this. Okay, actually, let's talk about this a little bit. So let's go up to here where we have the graph. Without bisphosphoglycerate in your blood, this is what hemoglobin acts like. It acts like it's essentially in the high affinity state all the time. So first of all, with none, you're not going to survive. Okay, but what it does is If you go to places where there's less oxygen, let's say high altitude, and you're there for a few days, your body will make more 2,3-bisphosphoglycerate, and it will take that high affinity state, it'll favor the lower affinity state more, and that's beneficial, and I'll show you that with a graph in a bit. But first, let's talk about how it binds to hemoglobin. So here's a crystal structure of hemoglobin, and here is, I don't know if you can see it. Here, I'll make it really big for a second, and then I'll shrink it back down so we can look at it all at once. All right, so you see here, it's kind of blurry, but here's 2,3-bisphosphoglycerate in this hole, okay? Remember from your other pictures, which state of hemoglobin has the hole? The T state, right. So I'm going to shrink it down again. So 2,3-bisphosphoglycerate is going to bind in the hole that exists in the T state. All right, so remember, these things are dynamic, right? We have still pictures we're talking about, but hemoglobin's running around your blood, and at least if it's not held in a particular state, it's going to be going, it's T state, R state, T state, R state, T state, R state. It's going back and forth between the two. And then depending on what conditions are around, you can stabilize one state or the other. So if I've got a hole in this is the T state right here. And this is my R state. Okay, I've got my R state here with no hole. I've got my T state right here with a hole. I'm going T state, R state, T state, R state, T state, R state. I'm in the T state, and 2,3-bisphosphoglycerate sticks in the hole. Now what happens when I try to go to the R state? You can't go because you can't close down the hole because there's something in it. All right, so you can think about it, you're just plugging up that hole, and now it's actually sterically preventing hemoglobin from going back until it pops out. All right, so 2,3-bisphosphoglycerate is going to bind in the hole that exists in the T state, while in the hole, it prevents the hole from closing up. Thus, it's sterically inhibiting the T state, or the R state, I should say. Does that make sense? Okay, so why is this good? Let me pull it down because I know some of you are still writing. But I want to look at the graph. So let's look at this graph. And what this graph has on it is it has a bunch of little curves, okay, where we have, here's what happens if you have BPG is equal to 0 millimolar. Okay, so we have no bisphosphoglycerate in your blood. Actually, it's not quite as bad as what I showed you, but it's still bad enough to kill you, right? So you go to your lungs, you pick up to 100% if you're at sea level, and then you go to what the oxygen is in your tissues, and you try and drop off, and you don't drop any off. Right, so we have to have some of this T state existing. So, everybody done writing? Make this a little bigger. So then at sea level, this is the amount of BPG you have at sea level. All right, sea level concentration. And so that gives us this curve. All right, and we've looked at how much oxygen we get and delivery at sea level. In fact, it's actually marked here on this, right? You look at sea level, you go from, here's where when you get your lungs, if that's maybe 95%, and then you drop off in your tissues. This curve right here is, what is that? That's like 60, 65%, and whatever percentages those are, that ended up being 0.38 or 38% of carrying capacity, all right? Now you go up to, I don't know what 4,500 meters is, that Mount Everest, whatever that is, high up, right? We're at about 1,500 meters above sea level, are we? Does that sound right? Or actually, it might be 1,500 square feet. Not square feet, but feet. You go up high, all right? There's a lot less oxygen around, okay? As you go up higher, you're up on top of the mountain, and what happens with your hemoglobin there is it picks up to now, let's say that's maybe 85%, and then it's going to drop off at, if the concentration of oxygen in your tissues has not changed, it's going to drop off here, and now you're only getting a delivery of 30%. This is 30% here. Does that make sense, what we're doing there? Okay, yes.
How does altitude affect the concentration
Of oxygen?
of the BPG?
Well, your body responds to the fact that you're not getting enough oxygen.
And it's Le Chatelier's. Yep.
Well, it's not Le Chatelier's principle. It's your body. It's actually your body makes more BPG.
Okay.
It's a biological. Your body recognizes. I don't know what the pathway is. Recognize you're not getting enough oxygen and makes more BPG as a result. Okay. But I don't know what the mechanism is for that. So, but if your body does respond and make more BPG, so I'm going to call this. Let's call this mountain, probably a really tall mountain, BPG, mountain concentration. Okay, but once again, it's not a Le Chatelier, it's not a chemical process, it's a biological process, okay, of your body making more. And so now we're shifting the curve over, right? And so intuitively, you think that's worse, right? We're worse at binding oxygen, but we're actually better at delivering oxygen, okay? So what happens is you do pick up less oxygen, right? Here is what you're picking up at this altitude instead of there, right? So maybe that's 5 or 10, 7% difference there, but you drop off a lot more. See the difference there? So if we're looking at this curve versus this curve. This is sea level BPG. This is mountain BPG. Okay? So we pick up less, but we drop off a lot more to compensate for that. Does that make sense? That's based on the curve shifting over. And then they actually have the percent here. So now it's 37%, from here to here, 37% of the oxygen carrying capacity of hemoglobin as opposed to 30%, which is a lot better. Questions? Your body also does other things when you go to high altitude. It makes more red blood cells. Okay, we've got various things. And this is a fairly quick, a couple day response, and then making red blood cells takes even longer. So that's why athletes will train at high altitude.
Can you just summarize that one more time?
Which part?
The change from 5 to 4 to
Yeah, let me go and write it in words. So 2,3-bisphosphoglycerate. So 2,3-bisphosphoglycerate favors the T state at high altitudes. More 2,3-B, or I'm going to say BPG here. More BPG is made. Shifting the curve to the right. Okay, and by shifting the curve to the right, I mean we favor the T state even more. Okay, but in terms of our curve right here, as a result, Hemoglobin picks up less oxygen in the lungs. But it drops off a whole lot more in the tissues. And I'm exaggerating a little bit to make the point. Yes?
Can you explain to me why in high altitudes more BPG just comes?
Why there is more?
Yeah.
Your body just makes more.
Okay. Oh, and that's something you said you didn't know before.
Right. It's like when you eat food, your body makes insulin.
Yeah.
Right? It will release insulin it's already made. Your body, when you go to a high altitude, your body is somehow sensing that there's less oxygen around you, and so it will make more BPG to compensate. But I don't expect you to know that. That's kind of the biology of this. I want you to be able to explain on a molecular level what does BPG do. Mostly, so we spent a bunch of time on this figure, and I think it's a cool figure. I would probably not ask you about this figure. If I did, I would give you the figure. I'd give you the other figure probably and ask you to tell me what percentage, read the figure and tell me what percentage of hemoglobin is being dropped off, right? I would not give you this figure because if I could get this figure with other things, maybe I'd ask a question about that. But that would be can you do this sort of thing, right? I'm going to ask you how it favors the T state, but full explanation of what's going on here. Certainly nothing beyond what I believe. Okay.
And then why does it pick up less? Like, why is it as a result less?
It picks up. So if we shift the curve to the right, we're just naturally going to pick. We've got a curve that. So I've got a curve that does. The reason why this works is because we're in the really steep part of the curve. Okay, because the really steep part of the curve is hitting here, and the less steep part of the curve is hitting here. But why does it pick up less oxygen? If this is the curve, right, and I shift it this way, then where I intersect where I take my oxygen concentration at the bottom, where I intersect my curve is a lower value, right?
Okay, thank you.
Anything else? Okay, I'm going to move on and talk about antibodies. All right, antibodies. Antibodies are an important part of your immune system, but I'm going to spend my time talking about how we use them as molecular tools. Okay? So antibodies are molecules that are made by the immune system, and your immune system, other organisms' immune systems. Okay, and they're bind to antigens. So let's go ahead and define antigens. I mean, antigens, this is going to be a circular argument. Antigens are molecules or functional groups we can talk about, let's say, or combinations. That are bound by antibodies. Okay, so it's another word. It's a fancy word for ligand, right? Antigens is a fancy word for a ligand, but in this case, well, it's only a ligand for antibodies. So these are made by your immune system. So if you have a bacterial infection, one of the things your body is going to do, it's got a whole bunch of different things it does as a response, but one of the things it's going to do is it's going to make antibodies that recognize or bind to specific things on like the surface of those bacteria. Okay, and that helps the immune system kill them. A whole bunch of different responses there. This is not an immunology course, so I'm not going to go into them in detail. your body, but one cool thing about this is what your body does, the way your body makes these antibodies, it just makes a bunch of random molecules, okay? I haven't told you what's going on, but let's go for this. Let's talk about this right here. I've got these two pieces of the antibodies, or they're right here, okay? This is the variable region, okay? And then the rest of this, you can see how there are C's next to them. The rest of this is this constant region. Okay, and so this is the same for all antibodies, at least of a specific type of antibody, all right? And then for that specific type of antibody, these are random and different between all of them. Okay, that makes sense? So what happens in your immune system is actually the DNA sections for immune cells get kind of shuffled around to make a randomization of these things. And then when your body's trying to make a set of antibodies, it makes a set of cells, each of which makes a single antibody. So this is the same for all the antibodies that cell makes. These regions are, okay? And then what their body does is it checks those cells against you. It kind of samples it. And if any of those cells binds to something that's you, they die. So it's like 2% or something of the cells that are made to make antibodies actually make it through. So anyway, that's how these sorts of things get generated. So at least your first generation of antibodies that you would use for a bunch of the different tests we're going to be talking about here, you're going to be exposing to an immune system to make them. You're going to take a rabbit, you're going to inject it with something. And then if that rabbit makes some antibodies you really like, you might extract immune cells from that rabbit and then use them to make a bunch of that specific antibody. Okay. Or it might be that you don't care that you have a specific antibody. You might just take all the antibodies out of the rabbit and use them. All right. Questions so far? So we had antibodies. You've just randomized the heck out of this sort of stuff, and essentially you can make molecules that bind to anything. All right, which makes them really useful when we talk about biology. And so one example of how you've used this, you guys have probably all used this test, right? So we've talked about this test already from the perspective of the gold standard DNA test, which some you may have had at the beginning of the pandemic. People were doing these, the gold test, but this is actually, when you do one of these, this is an antibody test. All right. So the COVID tests use a sandwich antibody assay or a sandwich ELISA assay. We'll talk about that. All right. And so how does that, and what they're doing, if we think about the COVID test, actually, before we talk about how it works, so the antibodies in that test bind to the nucleocapsid protein of SARS, what do I call the virus? SARS-CoV-2. Okay, so they bind to it. So what we have is it has an RNA genome, sorry. It's an RNA genome, and the antibody test is going to bind to this stuff. So you stick the swab in your nose, take the swab out, you stick the swab into whatever that liquid is. That liquid breaks up the virus, and these nucleocapsid proteins are now free floating. And then you detect them with antibodies, and I'm going to show you how. Questions? Okay, so the COVID antibody test that you take at home, because if it's a PCR test, you're sending that off. You're not doing PCR on one of those little kits. The COVID antibody test that you take at home detects the nucleocapsid protein. And I'm not, off the top of my head, I haven't looked it up recently, what we look for in the RNA genome. Um, that might be the DNA gene for the spike protein, but I'm not sure. Why they're probably looking for the nucleocapsid protein, you see there's a lot of it, right? This is the same thing. Each of these is a nucleocapsid protein, the same thing over and over and over. So this virus is going to have a lot of those little guys. It's probably the easiest thing to detect. All right, so let's talk about how an ELISA assay works. I'm going to walk you through it, and then what we'll do is then I'll write and walk you through it again. Okay? So we've got an antibody. That antibody has a target that it's going to bind to. Okay, it has 2 binding sites. And so let's say this is our nucleocapsid protein. We've got an antibody that can bind that. Well, let's have a second antibody. That antibody can also bind to the analyte. Okay, so it's not really shown well here, but down here you can see it. That can also bind to the COVID nucleocapsid protein, probably at a different site, because what we're going to do is we're going to bind it. We're going to have an antibody, we're going to have an analyte, and then we're going to have an antibody. That's why it's called a sandwich assay. Okay, the nucleocapsid protein is I'm vegetarian, so it's the cheese on your sandwich, and then the antibodies are the bread. Okay. So let's go ahead and look at this bit here. Let me go ahead and expand that so we can really see it close. All right, so you have a sample. This is a sample from your nose where you've broken up the various things, right? And then you put it on top. You're going to wash it over a substrate. So here we've got antibodies stuck to a surface. These are antibodies stuck to a surface that can bind. Don't think about the strip from the perspective of strip. Pretend we've got a little test tube we're doing it in for now. I will show you how this happens with a strip in a minute. So pretend this is the bottom of a test tube. So you pour the stuff into your test tube. If you have the nucleocapsid protein for COVID around, it binds to your antibody. All right, so now we've got it bound there. And so then I wash it. Okay, well, here's the wash. So I've got this. I wash it. So I wash off everything that's not bound. So now I have an antibody and I have my nucleocapsid protein bound. All right, then what I'm going to do is I'm going to add my second antibody. So here is my second antibody here. And that second antibody can bind to a different site on that nucleocapsid protein. So now I have my second antibody is bound to my test tube, or the bottom of my test tube, through the nucleocapsid protein, which is bound through that first antibody, the primary antibody. Does that make sense? All right. They don't show a wash here. Then I wash. I wash away everything that isn't bound. Then for this particular picture here, what they got is they have an enzyme. There's lots of ways you can do this. A tremendous number of ways you can do this. You have an enzyme that is bound to the antibody that's stuck to your test tube through the nucleocapsid protein through the primary antibody. Okay? And then this enzyme, if it's present, you add some stuff, and that stuff will either change color and be fluorescent, right? So we had a substrate. I guess our substrate's probably gray. If the enzyme's present here, it will make the pink stuff. Okay, and we detect the pink stuff. The pink stuff could be a color change, and we just do it with UV vis. It could be a fluorescent molecule, and we look for fluorescence. It could be, this is done all these different ways. It could be phosphorescence. So in other words, you put it in, and it's like fireflies. You put it in, and then it will glow, okay, on its own if you put it in the right reagents. It also could be, instead of an enzyme here, you could just have a fluorescent molecule attached. So all sorts of different things can be used. Yes?
So do you have that like orange part only if you're positive for COVID or if you're positive and negative?
This is the orange part?
Yeah.
If you're negative for COVID, okay, what happens is you don't have this here, right? This doesn't bind. If this doesn't bind, this can't bind. And then over here, you don't have, all you have is the green antibody. And when you put in your gray substrate, whatever that is, it doesn't turn pink. And pink is the thing you're detecting. Yes?
So are both antibodies like in the solution that you add the sample to, or like are the antibodies already in your sample? Like,
You're thinking about the exact COVID test?
yeah, or just this like is
So this is something that you might do in a if you're doing this research, you might do this in a 96 well plate. I don't know if you've seen those before. They're plates with a bunch of little wells. It's like 96 test tubes in this size. Okay, people have 384 well plates, so it's 384 test tubes in this size. There's all sorts of fun things you can do. You probably you have robots involved at some point here, but. So are all things involved in the thing I add? No, in this case, I'm adding several solutions. So I have my test tube's empty. Okay, let's say it's empty. I add this sample. Now I've got this sample in there. I pour out that sample, right? I wash it with something else. I pour that out, okay, to get to here. Then I add this antibody attached to a dye, enzyme, something I can detect. I then pour out, and then this stays stuck. If you had the COVID antigen, there's not a wash here, but then I'd wash it again. I had a wash solution. I'd dump it out, and then here I would add my solution that gives me a color change, a fluorescent molecule, something I can detect, and all those things I mentioned get done. It just depends on who made the test, what's best for them, that sort of thing. Okay. For your COVID test, what you're going to have is some sort of reaction. If you have a reaction, it's a reaction that's changing color, right? Because it creates a color, because you're looking for a color or a line you can see with your eye. Questions? Let's go ahead and look at that test, and then we'll come back and describe. I'll write this down. I'm going to give it to you. I'm going to do a COVID. This is how your COVID test works, but I'm going to do this is an animation for pregnancy test, which is also a double line test. At least so I'm told. All right, so this is going to be, I'm just going to silence it. Okay, so what we've got is a pregnancy test. The way a pregnancy test goes is you pee on a strip, right? And then stuff soaks up the strip. Urine soaks up the strip. And so what you're trying to detect in that is there's a molecule called HCG that's produced when you're pregnant. It's a hormone, I forgot what it is. But you've got, if you're pregnant, you get this hormone that's produced. And we're going to try to detect that. So instead of the nucleocapsid protein for COVID, this is the orange molecule that gets stuck between the antibodies. So on this strip, on this particular strip, so for a COVID test, you're actually you can have reagents in that solution you pour at the beginning, right? But this strip, you're just going to put urine on it, right? So your urine doesn't contain the antibodies to do the test. So that stuff is here. All right? So here we have these are antibodies that have an enzyme on them that's going to cause a color change. That makes sense? So this is the secondary antibody. Primary antibodies are going to be over here. So here we have primary antibodies. Okay, this is the test as opposed to our COVID where I think the test is lower. Here's the control. Okay, so these are antibodies that are going to bind to the molecule you make when you're pregnant. These are antibodies that just straight out bind to those antibodies. All right, so that's how we get our control. So here we're going to have a positive test, so that HCG, yeah, there we go. HCG hormone is present. It flows up the strip. It binds to the antibodies on its way up. So these are the antibodies that detect it. This would be our secondary antibody, actually. So the order is a little different. And this is going to get carried up the strip. Our narrator must be saying more. All right, gets carried up the strip to where we got the antibodies. So this is going to be this was our test, right? So we're going to capture these antibodies that have the enzyme that can be detected here. And then this part, this is our control. We're just going to capture antibodies by that constant region. All right? And the purpose of this is make sure your antibodies are still good, right? Make sure all your reagents are still good. So we've captured here and we've captured there, and now each of these have that enzyme on them, and they can take this thing and make a red color. They work with a reagent that is colorless, and they turn it red. And so by doing that, that's how you, for a positive test, end up being, well, why did we have, I don't know why they had two there. You should have both go, right? If you don't have both light up for your positive test, or both a red band for the same thing with your COVID, if you don't get the red band or the band for each, something's wrong if you don't get the control, and you shouldn't trust the test. All right, so this is a negative test. So here we have no HCG, right? So all these guys just run past. They can't bind to these antibodies. But if your test is still working, you should have that control strip light up, because we have antibodies that just bind to the constant portion of those antibodies. And so here we get our control is going to give us the red color. But here we have nothing to react with whatever that molecule is that's going to give us the red color. So there's our negative test. Questions? So your COVID test is going to act in a similar manner, because we also have the strips there. The reagents are going to maybe be present in, you can put more stuff into the reagents that are going on. But the strips themselves are going to have primary antibody. When we think about that, we think about having that strip, you have primary antibodies that are going to be present in both locations. Questions? All right, shall we go up and then talk about this ELISA? Let's go ahead and enumerate what's going on here. So let's talk about steps here. So step one, what's going to happen is a sample, in this case containing, so for this example, the sample contains the antigen to be detected. So we can think about that as, in this case, the COVID nucleoplasmid protein. Okay. A sample containing the antigen is added to the, I'm going to call it a test tube. Okay. It's not a test tube, but you do this in a 96 well plate or something, but added to the, I'm going to call it a test tube anyway, containing the antibody bound to the test tube surface. I'll just call it a tube. So that's the surface of the tube we're talking about it. Okay. And in that process, the antigen binds to the primary antibody. All right, second step, we're here on wash. I'll go ahead and put numbers next to these after I'm done typing. Second step, we're on this wash. Can everybody see it? Should I scroll it up a little bit? Okay. The tube is washed, and everything is removed except for the antigen bound to the primary antibody. Okay, that's our second step. Our third step is a secondary antibody with some way to make a color change, with some label, is added to the tube. So that label could be a fluorescent tag. So we could just directly see this. Enzyme, something to detect what's going on there, is added to the tube. If present, and it is in this example, the secondary antibody will bind to the antigen Okay. Questions so far? All right, 4, not shown. Okay, the tube is washed. And everything not bound to the antigen is removed. Okay, I'll label that in there as well. So we've washed this. We end up with this being clean. That'd be like right here. And then a reagent that will cause a color change, or that will react with the label to cause a color change, is added. In this case, react with the enzyme on the secondary antibody, or attached to the secondary antibody. The reagent is changed into something that can be detected. Okay, questions? Put my numbers in. I realized I forgot to plug this in, and it doesn't always give me a lot of warning. I want it for the next 7 minutes if I can get it. Okay. All right, so let's go ahead and put the numbers on there then. So the numbers for what we were talking about, we're going to add our reagent, we're going to wash it, we're going to add our secondary antibody, okay? We're going to wash it again, that's right here, and then we're going to add our final reagent that gives us a color change, it causes a fluorescent molecule, it causes it to phosphoresce, all sorts of different things can be used for detection. In the old days, we would have had a radioactive label on it. Yes? Scroll down? Yeah. I'd love to scroll down. And I'll put that up at the top. Oh, hold on, I'm going to scroll down a little bit more, and even a little bit more, then I'll scroll back up. And I'll leave the link in there in case you want to go watch the pregnancy test again. So this is in terms of antibodies. What I'm really going to want you to know is basically what is an antibody, right? It's made by the immune system. It can bind to anything. And I'm going to want you to know this ELISA sandwich assay well. Okay? And I'm going to talk in the next 7 minutes about a bunch of other things. I'm not going to test you on how they work, but I want to give you a feel for all the different ways that people use antibodies. They're really important tools. So any questions before I go into, here's a bunch of other ways I would use this. Yes?
So, does the label always bind to the secondary antibody?
Is the label always on the secondary antibody?
Or yeah, like
Yeah.
how does, so then wouldn't it always show up positive then if it always bound to the secondary?
So good question. So this is my secondary antibody right here, right? This is the primary antibody. The label is always bound to the secondary antibody, but I have this wash step here. So you see this guy?
Mm-hmm.
This guy goes away in the wash step, right? Because it's not bound to any antigen.
Oh, I see.
Right. So if you didn't have the nucleocapsid protein or the HCG hormone, right, this would be blank. There'd be nothing to bind to, and it would all just wash away.
Okay.
Other questions? Okay, then let's go ahead and talk about other ways you might use this. All right, so we got these molecules that bind to any other molecules, and we can take these antibodies and we can put labels on, right? We can detect things. So one way to use this that's really useful is it's called a Western blot or immunoblot. And the way this works is you have a gel. So we've seen these gels before, right? I have a gel, and let's say I have the gel and I've got some protein that is about this molecular weight, and I want to see it, and I want to see how much of it I have. And I run my gel, and my lanes look like this. So maybe that's it, right? I don't really know it's it, right? It's a pretty big band. There could be some, it could be in there, it could be under something. This could be my protein of interest, or it could be that it's not my protein of interest, it's something else. So it's hard to see where things are here. But antibodies, not only can antibodies bind to anything, but they're very specific, right? That's the whole point there. That antibody binds to the nucleocapsid protein of COVID and pretty much nothing else. All right, if they made their tests work well. And so the way to make sure that, hey, is this really my protein here, or is my protein really in there, under there somewhere, is I could then take antibodies and wash them over the surface of the gel. And if those antibodies have those antibodies that be attached to an enzyme or something as a label, I'd wash them over the surface, then I'd wash the gel to remove out everything that isn't bound. And if my protein is here in the gel, and that protein has an antigen I've got an antibody to, I could then get that to, we'll call it light up, right? So I'm looking at fluorescence there, I'm looking at radioactivity, I'm looking at all sorts of different things I could do. All right, and so the way that looks at the molecular level, so remember it's called a Western blot. So there's Northern who came up with the first type of antibody bot. No, Southern. Southern was the guy's name who came up. His group came up with the first antibody blot. And then I think after that, then they had Northern blots and Western blots. They kind of ruined the whole. It was this name. But anyway, this is how it happens. You've got a gel that contains your protein. All right. You could add an antibody that directly bound your protein and had a label on it. But more often what they'll do is you'll add an antibody, and then you'll add an antibody that just binds to all antibodies that has a label on it. And then you could have this sort of detection again. You've got a substrate that's going to go off of that label. Does that make sense? Why do you do two? It's really, that's a great question. It's convenient. So if I generate antibodies, like a rabbit, and I pull out a bunch of antibodies for this protein. I inject the rabbit with this protein. The rabbit reacts to the protein, makes a bunch of antibodies. If I don't want, I could then apply this antibody, and then if I have, let's say this is a mouse antibody, so it has the same region out here. All of them have the same region out here. If in my freezer I already have a bunch of anti-mouse antibodies already labeled, I don't have to label this with whatever enzyme that is. So conjugating an enzyme to an antibody is a difficult process. This would mean I just have to make an antibody, and then I could come and go in my freezer, pull out my anti-mouse antibodies, and just add those for labeling. Yes?
So it's just like in the assay where the partner antibody, it's like the secondary antibody, like it's like the last thing but kind of shoved together, right?
It's like the last thing, but it's not a sandwich assay where the antigen is sandwiched between the two. This is a matter of convenience. You could have that label directly on there.
Okay.
It would just be you'd have to do a new labeling conjugation every time you did a new antibody. so that this is more convenient. All right, I have 1 minute left to show you one more. All right, so let's say I had some protein in solution and I couldn't manage to purify it through chromatography or any of these other techniques. Well, one way I could do it is if I had an antibody to that protein, I could put the antibody into solution and then have a molecule that would help me spin that down to the bottom of the tube. So the antibodies stick to stuff, and then I have some way of making it big and centrifuging it down. Okay? And I'm out of time. But I had more slides. You could look at them if you want. But there are more techniques. Antibodies get used a tremendous amount in molecular biology.